Genentech as a bioinformaticist, chief bioinformaticist at Genentech. So without further ado, Zemin, go ahead.
Sunny, thank you very much.
I should say he pioneered the study of microenvironment of cancer using single-cell genomic approach.
Well, lots of people have done that, but thank you very much. It's a great honor to have the opportunity to speak in this exciting forum. I will first describe some of the bioinformatic tools developed in our lab. Then I will mainly focus on describing how we are applying the single-cell technology in understanding the tumor infiltrating immune cells. So first, we all know by now, the tumor is a fairly complex tissue with the tumor microenvironment influencing many tumor properties, including how they respond to therapeutic treatments. Inside the tumor microenvironment, or TME, there are many cell types ranging from fibroblasts, endothelial cells, parasites, to different kinds of immune cells. So this is where the immune cells recognize and combat cancer cells. So in a way, this is a battlefield for cancer immunotherapy. So we have learned in recent years that those tumor infiltrating immune cells possess special features that are very distinct from those located outside of the tumor. And different tumors exhibit different features. But it's not really clear what really determines the TME diversity. The complexity of the tumor microenvironment determined that single-cell technologies are needed to fully understand TME. So in single-cell sequencing, either SMART-2-based approach or 10x genomics technologies can be powerful in multiple types of analysis, including real cell type identification, tissue composition, so cell-cell interaction, marker identification, and cell lineage tracing. Before I get into the immunology aspect of the work, so let me first point out that many informatic challenges still exist in single-cell data analysis. So many of the analysis still don't have any well-established or standard tools out there. So for that reason, in the past few years, we have spent some energy, or lots of energy actually, in developing bioinformatic tools for single-cell data analysis. So here, I just want to quickly list some of the tools we developed as an advertisement for the tools we have here. So SFCC for clustering, iStarTrack for visualization, Rogue for cell purity analysis, CSO-map for spatial distribution and interaction, the CyBat for cell identification, IDEA for isoform detection, and IMAP, which is unpublished yet, is a cross dataset normalization method, and StarTrack is for T-cell lineage tracing. For today, I just want to quickly mention Rogue and CyBat that published this year. I think are fairly interesting. It has some conceptual interesting aspect. So, when it comes to clustering analysis, very often different people may have different results. For example, if you take this clustering results, some people may show this is 2 clusters, and some other people view this as 3 clusters. Some people view this as even 4 or 5 different clusters. So there's no standard qualitative metrics for describing a cluster. So what we want to have a standard way to do this. What we did was that based on the work by Chen-Wei Li and Pauline Liu, they found out that if you take a pure cell population, the mean expression for a given gene is actually forms a really linear relationship with their expression entropy. So this dot means 1 gene. But if you start to have heterogeneous cell population, some of the genes start to drop down from this really linear equation. So in fact, you can quantify the number of genes down here to describe the degree of heterogeneity. Okay, so in fact, you can derive this Rogue index, which is stands for ratio of global unshifted entropy. Then when Rogue is close to 1, that really means this is close to the pure population. Otherwise, it's not really pure. Okay, so you can use this to reanalyze some of the published results. But this is a results published elsewhere that we think this is described as 1 cluster, but has still a low Rogue value. Then we can reclassify this into 3 different clusters, which give more meaningful results. And also based on this concept, and we can derive the most distinguishing genes for a given cell population. In fact, we can construct what we view as a multinomial model for each of the cell clusters. Okay. Then for an unknown cell type, we would use a maximum likelihood estimation to quickly calculate what's the likelihood it belong to one of the known cell types. In fact, because this is a linear calculation, the speed of calculation is really blazingly fast. In fact, you can calculate about 100,000 cells per second. So it's a lot faster than the common tools like SCMap and Seurat version 3. In addition, for most of the tools, we show the accuracy are actually better than Seurat version 3 and SCMap. So this is a fairly interesting tool. And in addition, it's also useful to compare the given study to the previously published study. So for that reason, we collaborated with the company Analytical Biosciences, which is, I have to disclose, I'm the founder and scientific advisor for the company. So we essentially use a large number of people to collect all the previously published data, to organize the data in a way that's more meaningful and is more analyzable. So here we can think this is the largest single cell database in the world. It has over 1,000 data sets representing 20 million cells. So you can find the data, analyze the data, and integrate the data. What I really want to talk about today is actually to use such technology and tools to understand tumor infiltrating immune cells. So before I get into our unpublished work on pan-cancer analysis of tumor infiltrating immune cells, let me first briefly review our published work on infiltrating T cells to set out the background for our new projects. So we started by sorting out different types of CD4 and CD8 cells from liver cancer patients and applied the SmartSeq2 technology. We found that tumor infiltrating T cells possess characteristics that are distinct from T cells from either blood or adjacent normal tissue. We also use a combined expression and TCR-based analysis to show that developmental connection among T cell subsets. So this is liver cancer. Then we quickly did a similar analysis for lung cancer. Again, we identified more cell types. We also found that some of the therapeutic targets, including CTLA-4, PD-1, LAG-3, CD27, even though at the surface they are all labeled the same, but at the single cell level, they have very different properties. So this actually gives us a clue that the single cell data can be very useful in prioritizing drug targets. And we also did analysis of colon cancer. In this case, we identified additional cell types, and many of those have very distinct distributions in different locations. Now, okay, after we combined obtained the single cell data sets from colon, liver, lung cancer patients using the identical experimental procedures, we're provided with a unique opportunity to compare and contrast T cell properties across different cancer types. So when we examined the T cell composition in the blood samples only, we found that their differences were actually very minimum. But when we can examine the T cell composition in adjacent normal tissues, we found the dramatic differences, indicating that tissue location is a major determinant for T cell properties. Now, the tumor infiltrating T cells seem to harbor an additional layer of distinction on top of those tissue-based properties. So it seems like T cell properties are created by the combination of tissue, location, and cancer types. And such T cell differences are actually can be meaningful. We found out that liver cancer, colon cancer, higher presence of exhausted T cells in lung cancer, as well as activated Tregs. But in lung cancer, there's a higher abundance of the type of cells that we label as ZNF683 positive CD8 cells. This is what we view as pre-exhausted T cells. The abundance of pre-exhausted T cells appear to be associated with clinical features such as patient survival. We found out that those lung cancer patients with higher fraction of pre-exhausted T cells versus exhausted T cells had the best prognosis. By contrast, lung cancer patients with higher fraction of exhausted T cells versus pre-exhausted T cells have the worst prognosis. The T cell properties also may influence how patients respond to therapeutic treatments. We also know that among colorectal cancer, only the microsatellite stable or MSS patients are responsive to anti-PD-1 antibody treatment, but not those MSS patients. But the underlying T cell differences were not really clear. Based on our single cell data, we found out that MSS patients have much higher fraction of Th17 cells. By contrast, the MSI patients have a much higher cell type we call it Th1-like cells. So we call this Th1-like cells, not Th1 cells, because they highly express interferon gamma and CXCL13, but their interferon gamma expression seems to be under the control of a unique transcription factor called BLHHE40. So higher level Th1-like cells and their interferon gamma production may be important for the anti-PD-1 efficacy. So to summarize all this published work, the T cells can be classified in multiple types based on single cell expression data. It seems like different cancer types contain qualitatively similar but quantitatively distinct level of T cells. Such differences can be summarized in this small table. And such differences may impact clinical parameters, including how they respond to cancer therapies. So where do we go from here? So we obviously want to go beyond the 3 cancer types we already published and try to take a much broader view of the T cell properties across all cancer types. To construct a pan-cancer single cell transcriptome atlas for T cells, we essentially, this is unpublished work, and so we downloaded all possible published tumor data sets, including data sets with single T cells. But those are probably not enough. For those cancer types that are not adequately covered or not really published at all, we would go in there and try to collect new clinical tumor samples and generate new single cell data. So aim to get about 5 to 10 patients for each cancer indication. At the end, we collected over 300 donors, representing 21 cancer types. And after a series of quality controls, we obtained close to 400,000 high quality T cell data. Close to half of the data are generated by this particular study. So we use a combination of gene expression and TCR data to a series of downstream analysis. The problem is that many of the data are generated by different platforms, including 10x, MarSeq, InDropSeq, SmartSeq. So we obviously need to perform proper data integration to make the data more comparable. To achieve the data integration, we employed a 3-step pipeline. We first performed the Z-score transformation to ensure that all data sets are adequately mixed. We then grouped the cells into mini clusters in order to partially overcome the dropout problems and to make the data more stable. And finally, we applied the batch effect correction algorithm Harmony to further improve the data integration. So as you can see from the UMAP plots here, the cells from the step 1 to 2 to 3, cells are properly mixed, but the signals are become crisper. They are more crystallized. Okay, so at the end we have fairly good separation of the cell types, and we can use such metadata for data analysis. We call this cluster a meta cluster because they represent a cluster of a mini cluster of cells. Overall, we obtained a total of 41 metaclusters, and all these metaclusters are shared by at least 80% of all cancer types, indicating the cells are really well mixed. And each of the metaclusters has its unique distribution properties in tumor, adjacent normal, and blood. And so it is fairly obvious that the exhausted T cell shown here and the 4-1BB Tregs have higher abundance in tumors, but low in normal tissue and blood. So they have very strong tissue of cancer specificity. So there are a lot to talk about, but since this is a short talk, let me just focus on a few specific findings. And so exhausted T cells are of high interest. So let's focus on these cells. The exhausted T cell, or TX, of high interest. So we identified not one, but actually 4 exhausted T cell groups. Okay, they all highly express exhaustion markers such as TOX, CXCL13, TIGIT, CTLA4, and TNFR, SF9. But aside from the commonality, we found that two major clusters because there are high abundance of these two group cells. One is GZMK positive TX, and the other is terminal TX. Okay, they both highly express PD1, TIM3, and LAG3. And both highly express interferon gamma and coenzyme B. Those are effector molecules. But they're in the terminal exhausted T cells, they highly express lalin, ENPT1, and BATF molecules. Okay. So, but there are also that two relatively rare population. One is what we call the OX negative text. The other is TCF7 or progenitor text cells. The OX text here had the main feature that has a lower metabolism rates here, mostly reflected in oxidative phosphorylation and other pathways. So they are relatively inactive kind of cells. But TCF7 positive cells are more like stem cells. So they are commonly called the progenitor text cells. They highly express the genes that are related to lymph node migration, including CCR7 and cell. So next, we combine the gene expression and TCR data to dissect the trajectory of T cell exhaustion. So where do they come from? So I'm going to skip this part and directly go to the tumor aspect to see for exhausted T cell, where do they come from the naive T cells? So based on the gene expression similarity, based on using the multiple 3 tool and RNA velocity, we inferred two paths from naive T cells to exhausted T cells. One path that goes through the GZMK positive TEMs, and the other, and we call this is P1, and the other pass through the ZNNF 683 TRM cells. This is what we label as a pre-exhausted T cells initially. Okay, then we call this as P2 here. So, so in addition to expression-based approach, we also show that based on the TCR sharing, the terminal exhausted T cell strongly share TCRs with GZMK positive text and this ZNF 683 TRM cells, again supporting the development of path connection between these cell types. So primarily, the exhausted T cells mainly come from these two paths towards exhausted T cells. But if you look at individual cancer types, you see that different cancer types may utilize different pathways. So if you look at renal cancer, thyroid cancer, multiple myeloma, for example, they primarily utilize this P1 group. But for other groups of cells, including stomach cancer, HCC, they have more strong features connected to ZNF 683. But aside from this, some other cancer types, including breast cancer, they actually don't really have these main features connecting those two groups of cells. In fact, they are more connected to ISG positive CD8 cells, and other is connected to IL7R positive TRM cells. So even though some cancer types may have this exhaustive features, in terms of how they get there, may be different in different cancer indications. In fact, if you look at the presence of different type of exhausted T cells, you see dramatic different features here. So if you look at, for example, terminal exhausted T cells, which are more abundant in esophageal cancer, head and neck, liver cancer. But if you look at multiple myeloma, those are barely detectable, okay? And if you look at pre-exhausted T cells, those are more abundant in multiple myeloma, ovarian cancer, thyroid cancers, but very low level in renal cancer, multiple myelomas, okay? So clearly, if you want to derive strategies to target different type of exhausted T cells, you need to consider the relative abundance of this kind of cells in different cancer types. So based on the frequency of tumor infiltrating T cells, we define two major immune types. One show the high frequency of TEX but low frequency of TRM. The other one show the opposite groups. And these two major groups can be further divided into 8 subgroups designated from C1 to C8 here. So cancer types usually show enrichment of one of these immune cells. And such T cell-based immune cell typing provide sort of a new reference to understand the overall tumor infiltrating T cell properties. Notably, over half of the melanoma samples were of immune cell C2. Okay, that has high exhausted T cells, but low TNFR SF9 Tregs here, consistent with their tendency to respond to immune checkpoint blockade. Another example, the C3 groups is enriched in both thyroid carcinoma and uterine corpus endothelial carcinoma, uterus carcinoma. So C3 represent the tumors with low abundance of terminal exhausted T cells and high abundance of TRMs. Okay, again, the presence of these cells suggests these are the two cancer types that might also benefit from immune checkpoint blockade. Now, the T cell-based immune typing has a potential clinical implications. First, based on the TCGA large cohort analysis, tumors with with TEX low and TRM high tumor types tend to have better overall survival in both pan-cancer and individual cancer types, such as in renal cancer and liver cancer. Okay. Second, in the anti-PD-1 treated melanoma, tumors of the TEX low and TRM high tumor types tend to be responders. And the opposite, the tumors of the TX high and TRM low immune types tend to be non-responders. So to summarize this portion of the talk, we have developed various bioinformatic tools to accelerate and enhance single cell data. And we have used the single cell sequencing to identify and understand the unique T cell populations in various cancer types and review their dynamic properties. And different cancer types exhibit distinct properties of infiltrating T cells, which may have clinical implications. So we have just spent a lot of time talking about T cells within tumors, but T cells is just one major component of the immune cells within tumors. And other immune compartments can also be important and complex. And more importantly, we cannot study the particular cancer types in isolation. So it is more beneficial to study and understand the complete immune landscape inside the tumor. So in the next few slides, I will describe our recent work on the myeloid cell subtypes that seem to be specific in TME. So to this end, we have gone back to the liver cancer and used a combination of SMART-seq and 10x genomics approach for the single cell analysis. So we can allow us to capture a lot more cells. And we also include the lymph nodes and ascites in addition to the tumor and matched normal blood so that we can capture single cells across more locations. So I'm going to skip all the technical details and only present the final conclusion of the liver cancer study. Essentially, we painted the immune cell landscape in the different locations within the liver cancer. They are very distinct, which is a given. They are all different, but more importantly, they are more dynamic. Cells are not really stable and restricted in one location. There are some communications between different sites. So I'm going to give one example to describe this. Because we obtain a large number of myeloid cells, we are able to identify a group of cells that we call LAMP3 positive dendritic cells. What we found is that such type of cells were derived from CDC1 and CDC2 based on RNA velocity analysis. Okay, so they are enriched in tumor, so they got activated into a tumor, but they don't really stay there. So they are after they got matured and activated, they seem to leave the tumor and go to the lymph node portion where they will maybe activate other type of T cells. Okay, so so this is actually a very interesting subset of dendritic cell, which might hold secret to how tumor antigen signals are communicated. So we also identified a special subset of macrophage cells from colorectal cancer. So what we did was to use both SMART-seq2 and 10x genomics to survey the human colon tumor cells, and found that the myeloid cells are highly heterogeneous. Okay, in fact, we found 5 groups of macrophage cells here, and 2 of them enriched in tumor or seem to be tumor specific. For that reason, we label them TAM or tumor-associated macrophage. So these are the C1QC TAMs and SPP1 TAMs here, which I'm kind of showing here. So we focused on these two types of TAMs and found out that gene expression properties are actually very different. The C1QC TAMs here tend to express genes that are enriched in the complement pathway, the antigen presentation, and phagocytosis. By contrast, the SPP1 TAMs specifically express genes that are enriched in angiogenesis and metastasis. So this is also obvious if we project the TAM subset by expression of angiogenesis and phagocytosis related genes on the URD map. So based on all these, it seems like these two TAM sets might actually have very different or even opposite functions. One is pro-tumor, and the other is anti-tumor. So we should really keep this in mind when developing strategies targeting TAM. In fact, we used a random mouse model to study the effect of anti-CSF1R antibody, which is expected to deplete TAMs. So in fact, that it turns out that the anti-CSF1R antibody would selectively deplete only a subset of macrophages here, and shown here and here, okay? But it's leaving the SPP1 TAM intact here. So this is precisely the wrong outcome of the drug treatment. And this explains why the treatment was actually ineffective. So I have described some of the relatively new myeloid subtypes. From the liver cancer, we described the LAMP3 positive cells. And from the colon cancer, we described that TAMs are heterogeneous. In fact, we described C1QC TAMs and SPP1 TAMs here. And they have very different functions. So the question we want to ask next is whether these cells are universal across all cancer types, or they are specific to certain cancer types. To address this question, we again constructed pan-cancer single-cell transcriptome atlas for myeloid cells. So I'll skip all the details, in fact, that because I only have a couple of minutes left. So for each of the cancer type, we'll analyze separately and try to identify all cancer types. Okay, so here I just want to summarize. Then we perform the same analysis for all cancer types. In the bottom line is we're able to use consistent methods to identify all cancer types of monocytes and B cell macrophage. Indeed, we're able to identify LAMP3 DCs, C1QC TAMs, and SPP1 TAMs. So although their abundance may vary, but the cells are fairly universal to some degrees. But if you look at the master cells and dendritic cells, there are a lot of similarities that I'm not going to talk about. So if you look at CDC1 and CDC2 presence, they are fairly consistent. They're fairly similar as well. But if you look at the CDC2 derived LAMP3 DCs, there are a lot of variations, which I'm going to skip since I'm running out of time. So macrophage as well, there's a lot more diversity, but you do see the presence of these two types of cells in many of the cancer types, except that their relative abundance and the relative abundance in conjunction with other macrophage cells are actually very different in different cancer types. So since I'm running out of time, so I'm going to simply want to summarize that we have used single-cell sequencing to understand the detailed properties of tumor infiltrating immune cells. They have distinct properties from the cells outside the tumor, and they have dynamic properties. So, and different tumor types have possess different properties. So there's a lot of, so I feel like there is a lot of complications and mysteries. With the single cell analysis, many of the mysteries are starting to be reviewed. And this will help us understand the cancer biology as well as developing new therapeutic therapies, cancer immunotherapies. So this is just a very incomplete list of cell people I want to acknowledge, including the people who spend a lot of energy in the laboratory aspect, and as well as people who spend focus on the informatic aspect. I want to acknowledge our collaborators in the clinical aspect, as well as our funding resources. Thank you so much.
Thank you, Zemin, for the very insightful lecture on cancer immunology. Yeah, we have time for questions now.
Hi, Professor Zhang. I wonder why the different type of T cells distributed differentially among the different cancer types.
Yeah, that's a fairly interesting. It's a very fundamental question. Obviously, we would love to know the answers, but the answer is likely going to be fairly complex. So we suspect the tissue microenvironment will be a major determinant of the T cell infiltration. And we think it's not just the cancer cells that directly interact with T cells. In fact, we are under the impression, it's not really solid, but we think that the stromal cells, maybe different fibroblast cells, will possibly recruit different myeloid cells, and myeloid cells may further interact with T cells. But I think this is likely going to be a fairly complex process and procedure. And we are in the process of understanding this, and so far we don't really have the answer. But what we know in the general term is that tissue has a major impact, and the nature of the cancer cells themselves has a major role, because we may notice that the different cancer types of mutation may have an impact on the type of T cells inside the tumor. So, but this obviously would require fairly large amount of data, which we are still accumulating.
And another question is why you choose to research on the T cells first rather than B cell or other immune cells?
Well, T cells obviously are the central point. They are the focal point of cancer immunotherapy. And most of the cancer targets, immunotherapy targets, be that anti-PD-1, PD-L1, CTLA-4, TIGIT, LAG, TIGIT, ICOS, whatever. So all those target T cells. So essentially, those are direct players that recognize and combat cancer cells. So that's a more of direct players. So that's why we choose to use that to begin with. But we know by now that things are more complex. And other type of cells, including myeloid cells, we will have a more regulatory roles on this T cell. So that's why we cannot study T cell in isolation.
Okay, thank you.
Any other questions? Zemin, you mentioned the immunocancer therapy drugs only slightly today. Perhaps you could extend on the implications of this this pan-cancer to the immunotherapy treatment.
Well, I would love to. Yes, great point. And so the mentality we used to have is that you study one cancer type, you assume this is the same for all cancer. In fact, if that's the case, anti-PD-1 would work equally well across all cancer types. But we know that's not the case. But what is the reason? Why is that the case? Does tumor mutation load fully explain this? We know it doesn't. Okay, there's a more complicated answer than just the tumor mutation load. So only nowadays, when we start using single cell data, we know that the tumor microenvironment properties are very different across different cancer types. If you don't really understand this, we're guessing in the dark. We're just guessing, well, maybe the PD-L1 expression level can explain the efficacy. Okay, maybe the tumor mutation level can explain the tumor efficacy. I think those are likely all wrong, only partially right. But it's easy. That's easy. People easy to grab on something to say, oh, let's use this as a marker. I think going forward, we need more complex metrics to understand multiple parameters inside the tumor. I think we are only beginning to understand that inside any given tumor, there are T cell properties, myeloid cell properties that collectively determines the tumor phenotype. Okay, so I'm actually very excited with the ongoing the pan-cancer analysis, which is still ongoing. And so I think that would definitely give us a clue. So why something would be responsive? Renal cancer, for example, why would renal cancer be a good candidate for immunotherapy, despite this low mutation rate? Okay, I think this is something that people didn't understand. I think using the single cell study on the tumor microenvironment, we can start to answer these questions.
All right, that's an excellent note to end your lecture. Thank you, Zemin.
Thank you very much.
All right, Alexandra, hi, good to see you. The next talk will be given by